Okay, I think we can get started. Welcome everyone to the DFHCC Connect Science seminar series. And thank you for joining us. This is Cornelia Polyak from Dana Farber. And just as a reminder, we're recording the talks and we will post this at the website so you can look at later on if you miss it, with the exception if the speaker does not agree to recording. And also as a reminder, please send your questions in a Q&A function in Zoom, and the speaker we will answer as many questions as we can at the end of the talk, and he will have access to the questions later on to get back to you. And it's my pleasure to introduce Bernd Bodenmiller, who's joining us from Switzerland, where he's a professor of quantitative biology and also the chair of the Department of Quantitative Biomedicine at the University of Zurich. And Professor Bodenmiller did his postdoctoral training with Gary Nolan in California, and was a pioneer of using imaging mass cytometry for studying human diseases, with particularly focus on cancer. And he published some of the more seminal papers in the past few years using this technology to better understand the tumor ecosystem with particular interest in breast cancer, but also in other diseases. So thank you for joining us, and looking forward to your presentation.
Thank you for your nice words and for the nice introduction. So today I will talk mainly about the imaging, but the highly multiplexed imaging, but also I will present some of our previous results using simply multiplexed single cell proteomic analysis. So here you can see the survival curves of the molecular subtypes of breast cancer. I'm sure you all know how to read this plot. On the x-axis, you have the survival after the diagnosis in years. On the y-axis, you have the percentage of survival of patients falling within one of the four largest molecular subtypes of breast cancer. Now, what is very clear, if you look at this plot at a given time point, let this be here 10%, then about 80% of the patients are still alive and 20% have unfortunately deceased. Now, these plots you can do obviously of any type of cancer, and for other types they look even more dramatic. But the key point that I want to make with this plot here is that even if you look at the same subtype, the patients get the same treatment, you actually have a different outcome. In this case, this is survival. Of course, you can even more finely grained do stratification of patients, but it will not change that the outcome will differ. And the reason really is why. And Cornelia, she of course is one of the pioneers who made it clear that we all understand that heterogeneity is one of the main underlying causes. Since tumors can show cellular heterogeneity within a tumor, even within the same subtype, for example, of breast cancer, you will see cellular heterogeneity. That is something I will talk about today. But then also within a patient, once the tumor cells metastasize at the distant sites, or even the local lymph node metastasis, there are studies that show that here we have yet again a lot of heterogeneity of the phenotypes that you find, and that they also differ compared to the primary tumor. And it's really this heterogeneity that is why therapy response is different, and that's also why treating tumor and cancer overall, and you all know this, is such a big challenge. So clinical pathology, unfortunately, does not reflect this heterogeneity. A pathologist typically looks at the tissue, determines the cellular morphology, looks at one or few molecular features. In Zurich, we are now routinely doing genomic analysis of tumors. But ultimately, all of this leads to a very coarse grouping of the patients, and then standard treatments are used. And the question is really, how could we complement this, in fact, obviously highly valuable information, but how could we complement this and bring in more information into the clinical practice? So one way, and that's what we're working towards, is to look at tumors akin to ecosystems and then generate a comprehensive view of those ecosystems and try to identify patterns that then can be used for patient stratification and also to predict how to best actually treat a patient. And if I talk about ecosystems, then we should think about individuals. These are the different tumor cells that are within a tumor. These can differ. Then there are also there's an embedding, there's a soil in which these individuals are residing. That's the stroma. Then we also have normal cells that are further individuals, such as immune cells. You can have fibroblasts, etc. I guess in the audience here, you all know already about this concept of tumor heterogeneity quite a bit, and tumor ecosystems. So now, given that we look at the tumor as an ecosystem, what would we like to know about it to actually complement information that the pathologist gains? So first, we want to comprehensively know which cell phenotypes are present. We want to know the overall composition of the cells. We want to know where the cells are actually located and with whom they interact and with whom they actually communicate. Then we would like to know how this actually affects cellular function. And for every cell, we also want to know which pathways and functions are deregulated. We also would like to know about the 3D morphology, since in this example here, you see a blood vessel that will greatly influence the hypoxic state of other areas, and therefore it will also have a strong impact on the cell state and behavior. And ultimately, that means that we have to be able to perform molecular measurements that are spatially resolved in a 3-dimensional manner. So having such multiscale analysis can be useful, and I predict will be highly useful for clinicians, since it enables to find many scales that we can actually use to treat a tumor. And current treatments nicely reflect the fact that these ecosystem scales provide a broad repertoire of drug targets. If you want to target cells in breast cancer, you can do this with an anti-HER2 antibody. We can also target specifically signaling pathways. We can target the generation or development of 3D morphology, for example, by targeting angiogenesis. And of course, currently, what is of great interest to the cancer community is that we also have the ability to interfere with cell-to-cell interaction communication, such as by using an anti-PD-1 or anti-PD-L1 therapy to revert an exhausted to an active immune environment. So ultimately, what I hope one day we can achieve is that we comprehensively measure tumor ecosystems, that we can generate computational models that describe these ecosystems, and that we can use these models to make predictions about the best possible treatments. And therefore, that in the end, based on the reflection of the underlying molecular alterations that are present in every single tumor and patient, we can much more finely grained stratify the patients and provide a treatment that matches the underlying cause of the disease. And ultimately, having the ability to perform such measurements and analysis will, as I just said, greatly help to stratify patients, will help to make treatment proposals. And ultimately, and that's in the long run, always the most important thing to improve the treatment of patients is to actually understand the underlying biology. So in my group, we have a focus on breast cancer. We also work on other cancer types, but we have a focus on breast cancer. And despite the great improvements over the last decades in treating this disease, it is still an unsolved problem. 1 out of 8 women will actually develop breast cancer, and it leads to about 600,000 deaths per year, mainly through the development of distant metastases. The diagnosis of breast cancer really reflects the underlying tumor biology. Here are the molecular subtypes of breast cancer, the luminal A, B, HER2, and triple negative subtype. And these are evaluated based on the expression of hormone receptors. Then this is assigned to be a luminal tumor. And depending on the co-expression of HER2 or a high proliferative index, we talk then about the luminal B tumor. If there's only the expression of HER2, we have the HER2 positive subtype, and in the absence of a hormone receptor expression and or HER2, we talk about a triple negative tumor. As I just said, the treatment does reflect the underlying biology. So we use endocrine therapy for the luminal tumors to block the hormone receptor signaling, use anti-HER2 therapy, and for the triple negative tumors, since there's not one clear target, chemotherapy is used. And also for the luminal tumors, depending on the patient's risk, also chemotherapy can be used in case the patient has a high risk. Now, breast cancer historically was not considered to be a tumor having a high immune involvement, especially the luminal tumors are said, although now we and others showed it's not the case, that there's very little immune involvement. That is different in the HER2 and especially the triple negative tumors. And indeed, recently, the first immune checkpoint inhibitor therapy was approved for triple negative breast cancer. So now coming back to the aim to gain a comprehensive view on the tumor ecosystem, what do we have to achieve? What are our aims to do this, for example, in breast cancer? So first, we need to determine the overall composition of tumor ecosystems. You know, everyone talks about tumor heterogeneity, but very little work has actually been done to comprehensively describe cellular heterogeneity in tumors. Once we know about the composition, we also want to evaluate the overall structure of tumor ecosystems. And ultimately, we have to ask whether the information that we have gained about these ecosystems has any benefit or relevance for patients. And ultimately, that's what we want to achieve. And so in order to determine the composition of tumor ecosystems, we need single cell analysis. And as Cornelia mentioned before, I did my postdoctoral training in the lab of Gary Nolan at Stanford University. And there I was among the very first to work with mass cytometry. And we still use this technology in my group. I will explain you in a few seconds why. So here we take tissues, we dissociate them into single cells. We stain proteins with antibodies that carry a pure metal isotope as a reporter. Then we shoot those single cells into a mass cytometer. And as you know, the readout is the metal distribution and abundance for every individual cell. And since we know for every antibody to which metal isotope it was bound to, we can therefore infer for every cell the marker expression and phenotype. And routinely, over 40 proteins, phosphorylation sites, and transcripts can be measured on a mass cytometer with a throughput of about 1,000 cells per second. And we have also developed the tools that we can measure hundreds of samples a day. We just got the manuscript accepted. It's already also in bioRxiv, where we measured nearly 5,000 conditions where we did the drug screen against the breast cancer cells. And so this is possible. But the big strength here is that we can measure about 1,000 cells a second, because this now gives us the means to do an in-depth analysis of a tumor ecosystem. And we highlighted this in our first in-depth analysis of the breast cancer ecosystem, where we analyzed 144 tumors, of which 128 were luminal, and 50 control samples after dissociating the tumors into single cells. We then used 2 antibody panels, one focusing on tumor cells and the second one on the immune cells. And in the end, we measured the profile of 26 million cells across this patient cohort. The first question that we then had in this study was, how heterogeneous are now actually these breast tumor cells? And the answer is very heterogeneous. So here you see on the x-axis the different markers that we measured, and on the y-axis 45 distinct tumor epithelial cell phenotype clusters, and the color scale indicates from blue lowest to red highest the expression level of the markers that you can see here. In order to better comprehend the biology of these samples, we performed meta-clustering, and this allowed us to identify 2 basal groups and 7 luminal groups across those 45 distinct phenotypic clusters. If we then look a little bit closer, then we see here for these basal cells that we have high expression of keratin 5 and or 14 and vimentin. But then I apologize, I'm not sure why it went the wrong direction. But then what became extremely interesting is when we looked at some of these luminal meta clusters, since in this luminal group 6, we are of androgen receptors, of androgen receptor, and also very high level of PRB, and also at least for luminal group 6, high levels of estrogen receptor. And in addition, in this luminal group 6, we also had a high expression of BCL2 and survivin, and they provide anti-apoptotic signaling. And together with androgen receptor expression and other markers that we found, we thought, given the literature, we still have to prove this experimentally, what we're doing in the moment. But just based on literature knowledge, these two phenotypes, as we concluded, have a high resistance to treatment. And that's also something that already is known and has been showed for these more basal-like phenotypes. So what we then did with this data is we wanted to identify patient groups. And on the level of the meta clusters, I will show you in the third part of my presentation that we can actually do this. But on the level of the individual phenotypic clusters, we were not able to group the patients. And therefore, we thought that we need different measures to actually describe tumor heterogeneity. And so the first measure that we introduced is called tumor individuality. And across all of the cells that we analyzed, we build a graph. And then we asked for every single cell, are your neighbors in the graph cells from the same tumor? Or are the neighbors in your local environment, are the cells from other tumors? And if for a tumor, then the cells were mainly together with cells of the same tumor. We have a tumor that is has a high individuality. But if a cell or a group of cells comes from different tumors, and if the cells of a tumor are often next to other cells of other tumors, we have a low tumor individuality. The second measure that we developed is what we call phenotypic abnormality. And here we use the deep autoencoder, and we trained the autoencoder using the control normal breast epithelial cells. And what we can then do is we can run, for example, a normal cell through the autoencoder, or we can also do the same with the tumor cells. And then the autoencoder can provide us what is called an error, which basically says how different is the cell from the normal epithelial cells with which we trained the autoencoder. And this ultimately, thinking about the markers that we measured, simply means how different is the combination and abundance of the markers on an individual cell. And this then we call the phenotypic abnormality. And then we calculated this phenotypic abnormality for every tumor of our cohort. And then we ordered the tumors according to the phenotypic abnormality. And just to explain you this plot a bit better, then for every patient, we plotted the 45 distinct tumor epithelial phenotypes where the presence is indicated with such a bubble that you see on this bubble plot. And the bigger the bubble and the more it goes towards orange, the more dominant such a phenotype is within a tumor. And you can immediately see that not a single tumor is like any other tumor. So really any tumor is unique. When we then also looked at the phenotypic abnormality that I just described and plotted it against individuality, we saw that those tumors that are highly abnormal, they're also those that show the highest individuality. Or put differently, when we put the individuality versus the richness, and the richness is the number of the distinct phenotypic clusters, then you see that the tumors over here that have a high individuality have then a very low number of different tumor cell phenotype clusters. Finally, we then also saw that the phenotypic abnormality shows a relationship to the grade with the hormone receptor status and also with the molecular subtypes of breast cancer, and all the clinical features that are associated with poor and then even poorer prognosis show a significant relationship to phenotypic abnormality. The phenotypic abnormality really captures the higher it is, the poorer the prognosis for a patient. Finally, when we then looked into more detail at the phenotypes of those patients that are dominated by few of these tumor cell phenotypes, then we saw that these were the basal cell phenotypes, and this luminal 6 and 7 phenotypes that I just described to you before, likely having a higher level of resistance to treatment. And here we could conclude that these resistant phenotypes, that they actually dominate tumors and are associated with poor prognosis. And when we saw this, to us it was in a way not really intuitive, because we always assumed that the most heterogeneous tumors are those that can best deal with treatment. But it looks like that those tumors that have the poorest prognosis are actually those that are dominated by one or few tumor cell phenotypes. Then we also wanted to know whether we can find immune environment groups and what the overall state of the immune environment is across this whole patient cohort. And I will now only show you results that we found for the T cells. We also did an in-depth analysis of macrophages, but due to time reasons, I will only talk about the T cells. And in regards to the T cells, here we were really interested whether the T cells are inactive, whether we have activation that would be shown by a certain level, for example, of PD-1, or whether the T cells are actually exhausted. So here you have a cytotoxic T cell with a very high expression of PD-1 and co-inhibitory molecules such as CTLA-4 and TIM-3. So first we checked whether at all in our cohort, which is dominated by luminal tumors, we have T cells expressing high levels of PD-1. And indeed, comparing the frequency of these T cells in tumor versus the control samples, we saw that there was a significant increase. And we also saw a significant relationship, again, to poor prognosis as defined by the grade. Then we looked closer at the individual T cell phenotypes. Again, on the x-axis, you see the different markers that we measured. On the y-axis, the different T cell phenotypic clusters. And the abundance of the markers are shown from lowest blue to red highest level. And now we'll only focus on those cell or T cell phenotype clusters that actually express PD-1. And here I want to highlight especially 4 different T cell phenotypes. One that we call T11 expressed very high levels of PD-1, very high levels of TIM-3 and CTLA-4. So here we can be fairly sure it's an exhausted phenotype, as well as very high levels of CD38. That's over here. And we have in the previous publication shown, and others in parallel, that CD38 can also be an indicator of T cell exhaustion in the context of carcinomas. Then there's another phenotype, this T7, which expresses CD38 and PD-1. And here we assume that's a phenotype where we have activation and are already a little bit bounding towards exhaustion. This T14 phenotype, to us, given the markers, looked like being activated. And we also had regular Tregs in our data set, which have high level of CD25, FOXP3, and CTLA-4. And Tregs are also indicators of an exhausted immune environment. So then we also looked at the frequencies of these different, of the Tregs and this T11 and this T14, etc. phenotypes when compared all the tumors that were hormone receptor positive versus those that were hormone receptor negative. And you can see, for example, for the Tregs, that the frequency is much higher in the hormone receptor negative patient samples, but that there's a fraction of hormone receptor positive samples where we also have a high frequency of the Tregs. And the same is true for this T11 phenotype. It's in general higher for the hormone receptor positive, hormone receptor negative samples, but you also have a subset of the hormone receptor positive samples where we have equally high or up here even higher fraction of this highly exhausted phenotype. So once we saw that we have this continuum of T cell states across all of these 144 tumors, we then were wondering whether we can group the patients based on their immune environment. For this purpose, we plotted the frequency of all of the T cell and also macrophage phenotype clusters And then we performed hierarchical clustering. And ultimately, we could identify 3 distinct, what you call tumor immune groups. The first tumor immune group, that is what we call the TIC1. Here we basically have an inactive immune environment. Then we have the so-called tumor immune group 2. Here we see, based also on the macrophage phenotypes and this T cell phenotype and other phenotypes, that we actually have an activated immune environment. And ultimately, what we then also saw is that in this tumor immune group 3, we have a combination of these highly exhausted phenotypes. And that shows us, here I made a zoom in into which molecular subtypes of breast cancer we're looking at in this tumor immune group 3. You can see that we have a couple of triple negative, 1 or 2 classified tumor, but mostly we have luminal B high-grade tumors. And they showed us that among the luminal patients, we actually have a subset, it's 80% of patients with an exhausted immune environment. And that was actually, again, a big surprise to us, since it typically was assumed that luminal tumors do not really have a high level of immune activity. And here one could even hypothesize whether immune checkpoint inhibitor therapy could be used for the treatment of these patients. Now, finally, and that was actually something really fascinating to see, many of them plotted the phenotypic abnormality and individuality for these 3 tumor immune groups. Then we saw that this phenotypic abnormality, which was trained purely on the tumor cell phenotypes, this algorithm, this network never saw any immune cell, that we see a very nice and strong correlation to the tumor immune groups, and that the tumor immune group 3 with the exhausted environment is also, these are those tumors that have the highest phenotypic abnormality. Finally, we also wanted to know whether these tumors are actually structured. And what I mean with that, are there cell types that coexist in a tumor or typically exclude each other within the tumor? And indeed, if we make this correlation analysis here for all tumor versus all tumor cells, we see that typically the same phenotypes only coexist. So it's not a lot of mixing. That again reflects individuality, but that we see nice relationships of tumor to immune phenotypes and also of the immune to the immune phenotypes. And ultimately, this showed us that these tumor ecosystems are heterogeneous. So now I've shown you how we can use single-cell proteomic experiments to determine the composition of tumor ecosystems. But ultimately, we want to evaluate the structure of the tumor ecosystem. We want to know whether these statistical relationships are actually spatial relationships, whether there could be a mechanism underlying this observation or these observations. And therefore, what we really need is spatial cell analysis in situ. And to enable this, my group developed what is now called imaging mass cytometry. And in imaging mass cytometry, we take routine tissue sections on the routine slides that all the pathologies are using. Then we stain the tissue again with antibodies that carry these pure metal isotopes as reporters. Then we use a laser ablation system that rusters over the tissue. And every time the laser hits the tissue, it removes about a square micrometer of the tissue. And a single cell is 20, 30 micrometers in diameter. That means we have subcellular resolution. And every time the laser hits the tissue, it generates a particle cloud of the tissue. And these particle clouds, akin to the single cells, are then analyzed in the mass cytometer. And the result is that of every laser shot, we gain then for this area where it shot or hit, we know about the distribution and abundance of the isotopes in that location. And therefore, we know about which markers were expressed. And ultimately, such a laser shot is then akin to a pixel in an image. And in the moment, we routinely measure 40, sometimes a few more markers with subcellular resolution on tissues. The images that we can generate with imaging mass cytometry, they're really akin to fluorescent microscopy images. Here you see an MDA MB231 cell line, where in blue is DNA, in green vimentin, and in red you see CD44. And if you zoom in, you see that we can nicely see the vimentin filaments of individual cells. Over here, we have a cell that is in mitosis. Also, I don't show it, but If you look at phosphorylated histone H3, you really see the chromosomes as they are pulled apart. We did lots of validation experiments and can really show, here's a direct comparison of fluorescence microscopy to imaging mass cytometry, that we are very comparable to this technology. We have a little bit low resolution, you can also see that, but then we have the advantage of being able to do highly multiplexed imaging on tissues. Imaging mass cytometry also has other advantages over fluorescent-based approaches. Here you see a whole slide scan using fluorescence microscopy of a bone metastasis of a breast cancer patient. And you see in yellow pancytokeratin, in red CD68, and in blue you can see DNA. Now I zoom into this region over here, and we imaged also the same region with imaging mass cytometry. And several things become obvious. Here to the lower right or to the upper right, you see lots of signal in fluorescence microscopy, but an absence of signal with imaging mass cytometry. And here we have high levels of autofluorescence in the fluorescent measurement. Also, which you don't see here because it's zoomed out, there can be lots of tiling effects if fluorescent measurements. If you do serial fluorescent imaging, where you have to strip, stain, etc., you really have to optimize the antibody order. We also are doing this and in collaboration with other groups where we had to learn a hard lesson on that. And the dynamic range is a few hundred to about a 1,000-fold higher in imaging mass cytometry compared to routine fluorescent-based techniques. What we also developed due to the high number of markers that we can measure is the simultaneous detection of transcripts and proteins. And so to achieve this, we use a branched DNA approach where we build basically a Christmas tree of DNA on an mRNA. We then come in with metal-based imager probes, and we can also co-stain with the antibodies. And over here, you again see your breast cancer section. It's, I think, about 3 times 3 or 4 millimeters, where we looked at many different markers. And what we really use the transcript detection for is to determine which cells are producing chemokines, cytokines, or just in general growth factors, since they are secreted and therefore very difficult to detect. Also, the antibodies for those are actually not really good. I don't know why this moves on. And so here, just to zoom in, you can see here the boundaries of the epithelial cells based on pancytokeratin and ikaterin. Then you can see up here in green the cytotoxic T cells. And here in italics, you see CCL18, CCL5, CXL9. Those were measured using our mRNA probe. That's the same region and more markers, where we also then, where you can see imaging of CXL10, CCL2, and CXL13. And that's all the same image. So here we again have 40 markers, and 12 of those are actually transcripts. Finally, I talked also initially that we're interested to look at tumors actually in 3 dimensions. Because if you look at the tissue section, and if you want to determine cell state, well, you miss what is above and below a cell. So we miss a lot of information about what could explain a cell state. And so to enable 3D analysis of tissues with imaging mass cytometry, we developed a workflow where we have a special knife, where we do serial sectioning of tumors. We then optimize the protocols for the antigen retrieval, the staining. We perform imaging mass cytometry. And then in silico, we register the serial sections. We generate a 3D voxel model. And then we do a 3D segmentation. And then we can use the 3D data for all the single cell analysis algorithms that you actually know of. And this is a project that we do within the Cancer Research UK Grand Challenge. We are part of the IMAX team, that's headed by Greg Hannon, who is at the University of Cambridge. And in a part of our consortium, we also have a group that is building a virtual reality to actually walk through tumors in 3D. And it really works. It's amazing. I can really recommend you once to ask to get a demo to do this. Or at one point, yeah, you should really do this. Now, how do these 3D models look like? Here's an example of a HER2-positive breast tumor. And now you can see a voxel model where pancytokeratin is shown in yellow. In white, you see immune cells. And in red, you actually can see van Willebrand factor, so blood vessels. We then also segment those voxel models into cellular models, and that is shown here, where the HER2 positive cells, they are shown in orange. Vimentin positive cells, they are shown in pink, and SMA in light blue. And it's really impressive to, for the first time, see a tumor in such a high multiplexicity. So again, there are many more things that I could show you, but due to time, I can't. And it's really fascinating to get 3D data of tumors. And here's an example why 3D is relevant. To the left, you have a voxel model. And in a voxel model, we could see a region where the structure and the surface of the tumor was really not smooth. And then we saw after single cell segmentation that most likely we're looking here into invasive individual tumor cells that are emerging from a microprotrusion of this tumor. And so by pure chance, we are looking at the spatiotemporal process of cells going out of the tumor and entering its surrounding. And that is one of many examples that I could tell you why 3D analysis is actually highly relevant. Now, finally, given the data that we can generate, we also need computational tools. And my group develops various computational tools for the analysis of basically any type of multiplexed imaging data. You find all our tools on our Bodenmiller Group GitHub. Probably what is best known is the HistoCAD toolbox, where you can visualize images, you segment them, you can look at the individual phenotypes that are plotted onto the images. We, of course, also know what markers each of these cells is actually expressing. We also developed algorithms that tell you whether cells are more frequently next to each other, so that there's some structure in the tissue, or whether they avoid each other. It's also generating structure. I will show you later on how we also use it to define communities of cells. And this year also how we can define networks. So we call it the social or asocial in the case of cancer network of cells. You can also mask the tissues. Here you see in red the tumor regions, the stromal region in black and blue. You can calculate distances. So we have lots of tools. If you're interested in some of those tools, just let us know. And we are always happy to actually help. And another tool that we just published is the CytoMapper tool. And that is really meant for everyone who loves R, since then many of the analysis that I just showed you here can actually be done simply directly within the R environment. So another important point that I want to make is that in Zurich, we are also part of an observational clinical study where we get the patient samples directly after the operation, and then we perform suspension mass cytometry and imaging mass cytometry, and the whole process from the sample processing to data generation to data analysis to generating a report that then goes back to the molecular tumor board. We have a turnaround time of 32 hours to achieve this. And this was only possible since most of the steps that you can see here are actually fully automated. Now, just to summarize the second part of my presentation, the current status of imaging mass cytometry is that due to the mass spectrometry basis, it has a very high dynamic range. You can really analyze any type of tissue and sample as long as you can bind the antibodies. The limit of detection is similar for fluorescence microscopy. We have seen that the images themselves and the resolution is similar to routine fluorescent microscopy. One drawback is that it's a fairly slow technology. To analyze 1 square millimeter takes about 1 hour. That's still comparable to other multiplex imaging techniques. However, compared to other routine imaging techniques, that is actually slow. We can measure transcripts, proteins, modifications at the same time. You can just load your slides you get from the pathologists. We can do IMC in 3D. We also just published a paper where we showed a barcoding approach that allows 3D tissue screening, such as tumor spheres and organoids, and you can analyze hundreds of them in parallel. And most importantly, really, imaging mass cytometry by now is very robust and very reproducible. For the clinical study, we had to do lots and lots of reproducibility testing and validation. And at one point, I can just give a talk about that. And we could show that it's highly reproducible. And given that, and given also that we can measure 40 markers simultaneously, the size of the projects in my group, they keep growing. And in many of the projects, we now analyze over 1,000 patient samples. Because once you have, for example, a team A, you just load it, and then the machine is just running for a couple of weeks. You load the next team A, and in the end, you have the data for the analysis. And just the measurements now is, in the end, a very short part of a project. And data analysis, I would claim, is now the majority of what the people in my group do in the life cycle of their project. Okay, just quickly. Imaging mass cytometry, as you have seen, is also the part of this CRUK project. We are part of a very large European project where we look at 3,000 patients and the clinical trial settings. And so it's really a useful tool, I have to say. Now, continuing of what we can do with imaging mass cytometry. Now that we know the structure of these tumor ecosystems and their composition, we also want to know, is the data that we generate, is there any potential that it one day might be useful for patients? And here I want to show you a retrospective study where we went to a biobank. We got 281 breast cancer patients where we had associated long-term clinical data. And the data that we especially had here was the long-term survival data. And I showed you initially this Meyer-Kaplan curves. And our question here really was, can we perhaps better, more finely grained stratify the breast cancer patients? And to achieve this, we then developed an antibody panel that includes different cytokeratins, the standard markers used by pathologists such as ER, HER2, Ki67, markers of the immune context, mesenchymal cells, endothelial cells, transcription factors, growth and division markers, an epigenetic marker, to get really a comprehensive view now on the breast cancer tumor ecosystem. And overall, we analyzed about 1 million tumor cells. So then we first wanted to know, are those single cell phenotypes, how are they distributed across the molecular subtypes? And over here you see the luminal A, the luminal B, HER2 positive, and the triple negative subtype. Now, just looking at the many colors that you see for every subtype quickly makes the point that the current classification does not reflect the phenotypic heterogeneity. To analyze this then in more detail, we plotted for every patient the density of the tumor, immune, and stromal cells. So that's in the given column. The immune cells are typically green, stromal cells light blue, and all the other colors are the tumor epithelial cells. And then we performed hierarchical clustering, and you can now immediately see, just looking at this plot, that those colors indicate their groups. And indeed, we identified what we call 18 single cell pathology groups. Now, given the clinical data we had for this cohort, we wanted to know if we now draw the Meyer-Kaplan curves or plots for all of these groups. If it's random, likely it would be similar to the average of the whole cohort, which is shown here in black, or is it different? And the clinical subgroups, they're shown here in green. That's the triple negatives with the poorest prognosis, luminal B in pink. In orange, that's the luminal A tumors, and the HER2s, they are in this brown, bluish color. So then we did the same plots for our single cell pathology groups. And here you have mainly luminal A patients, and we could then identify a subgroup with an excellent long-term survival, while the other groups, although being luminal A, have now a poorer than what you expect for this molecular subtype survival. We also found this for the more luminal B tumors, again, a subgroup that has an excellent long-term survival. And even for the triple negative patients, we could identify one subgroup when none of the patients died, while then for the others, we had again an equal to poorer survival than what you would expect for this molecular subtype. So this means that we generated, or we found instead of 4, 18 distinct, what we call the single cell pathology subgroups. And that at the minimum, we could identify those patients that had a good prognosis. Now, the data that we generated here, what really is the strength, I would say, compared to, let's say, bulk transcriptomic or the bulk proteomic measurements, is that we have data that's very easy, but not easy, but it can be easily, more easily biologically interpreted than just a long list of proteins that are up or down regulated or transcripts that are up and down regulated. Because we have images of cells that we can associate with a biological meaning. And so we started to look at these images a lot to ask, why is this single cell pathology group 1 and the single cell pathology group actually different? Because if you look at the tumor cell phenotypes, then you can see that this single cell pathology group 1 is dominated by sheets of this brown phenotype. We also have this pinkish phenotype, and the single cell pathology group 3 has the very same tumor cell phenotype and also this pinkish tumor cell phenotype. So if you just look at the phenotypes present, they're the same, but now why are they different in their survival? And one thing we actually realized eventually was that the overall structure of the tissue was different. And so if you have such images, what we first developed and published 3 years ago is that we can study the neighborhoods of cells and the social networks. And others, for example, Oliver Stegle has a very nice paper where they show how you can analyze gradients across these tissue images. And we thought what we really would want to study is not just neighborhoods of cells, but whole groups of cells, whole communities of cells. And to achieve this, we then took the images, we generated a graph where each cell is a node and the neighboring cells are connected using edges. And then we used the Louvain algorithm to identify communities of cells. And all of you probably know Louvain indirectly very well, since on Facebook, I think they still use Louvain to determine actually who are your best friends based on the connectivity that you show in your social interactions. So then we looked at the different communities that we identified. First at the epithelial cell communities. And here you see on the x-axis, the average cells per community, on the y-axis, the communities. One thing was striking. If you again just look at the colors, these communities, they're fairly homogeneous. So there's very little mixing of different cells within these communities. And where they mainly differ is in the size. We had small, medium, and large sheet types of communities. This was very different for the stromal communities. Here we have all sorts of sizes, but we always have a mix of different types of the stromal and immune cells. And here we could really see that it's overall the structure of these communities that is driving the differentiation. And then we went back to our single cell pathology group 1 and single cell pathology group 3, and we compared the dominating communities. And indeed, we found that the single cell pathology group 1, that is from here to here, was dominated by a different community than the single cell pathology group 3, the poorer survival, which was dominated yet by a different single cell community, epithelial cell community. And we then took this information and we made calculated the hazard ratio model only using communities and also the molecular subtype. And then we could see that the single cell pathology group 3 is enriched for this community 17, it's associated with poor prognosis, and the other one with good prognosis. That's what you would expect based on the survival curves. But once we applied this algorithm, we saw much more extreme cases. Like here, this violet phenotype is dominating both of these groups, but if we only have small sheets, we have a very poor prognosis. While if we have these large sheets, we have a really good prognosis. And we found this over and over again. Now, what got really interesting, though, is when we looked at the stromal communities, then we found, before I told you about these triple negative patients that had a very good long-term prognosis, and they actually were dominated by those communities that are here associated by the good prognosis. And so, to summarize my presentation today, I hope that I could show you that although breast cancer ecosystems are very heterogeneous, we still identify clear patterns and relationships, and that those patterns and relationships on the level of the tumor and the immune cells enable us to stratify breast tumors and patients, that the experimental and computational imaging mass cytometry workflow enables reproducible multiplex tissue image analysis. I really have to say across thousands of patients, that we introduce tumor abnormality, individuality in those communities as novel descriptors of these tumors, and that also that those are predictive of patient risk. And ultimately, that the single cell pathology improves patient stratification. And one thing I would like to mention here quickly, if you have large clinical studies where you have two arms, treated, untreated, of breast cancer, we would love to connect treatment response to the single cell pathology groups, and please write me an email in case you're willing to share your patient cohort. And with this, I want to come to the acknowledgements. So first of all, I want to greatly acknowledge my group overall. All of this would never be possible without their fantastic hard work contributions, thousands of ideas, and it's just a fantastic group to interact with. And I greatly want to acknowledge Johanna Wagner. She led the first project where she looked at the 144 tumors in collaboration with researchers from IBM. And here I also want to greatly acknowledge the PATH Foundation, the biobank that was founded by patients to make sure the samples are made available for researchers. And they, at that time and now, continuously supply us with patient samples that we need. And I also want to greatly acknowledge Jana Fischer and Hartland Jackson. They led the last part of my presentation, the analysis of the breast cancer cohort where we had survival data. And in this regard, I also want to greatly acknowledge the University Hospital of Zurich and the University Hospital of Basel for providing us also continuously patient samples for our work. And finally, I want to acknowledge the Swiss National Science Foundation, the European Research Council, the NIH, and the Cancer Research UK and HubMap for being the main financial supporters of the work in my group. Really thank you for that. And with this, I'm done and I'm ready to receive your questions.
Okay, thank you very much for this great talk and beautiful pictures. I really like this. So we have a number of questions. Let's start with, there are a couple of technical questions related to the technology itself. So one of them asking if the, as we know, the quality of staining is always dependent on the antibodies. Yeah. So if you have, how do you differentiate whether it's, if you see differences between different tumors or different parts of the tumor, where that's really a real difference versus just technical variability?
So it simply means that, you need to have a lot of control samples that make sure that your technical variability is very low. And we have evaluated this thoroughly, and that the technical variability itself of the technology is actually very low. Now, a different issue is antibodies. And to generate a good antibody panel is a lot of work. It can take a person many, many months to identify antibodies that work robustly for the samples that you want to analyze. And for us, it's mainly FFPE samples. And to validate those antibodies, you need, again, control samples where you know what is expressed, where is it not expressed. Co-staining actually really helps to validate, since you know for certain markers that are allowed to be together or not. We do also ex vivo, for example, for the immune cells, we stimulate them so we know which markers we have to expect on cells. Then we also use them for validation. So it's actually a really big effort. What is challenging to control, though, is the quality of the tissue samples. Because there can also be quite some variability be introduced based on how long they have been in the formalin, how they were treated, how they were stored. And that is a bit more of a challenge. And there David Rim had some good ideas on how to control for that. But in this regard, also having certain controls, and especially having very high number of patients, and then do further validation after your findings on a different cohort from a different hospital, that's in the end what you have to go through to substantiate your findings.
Mm-hmm. Okay. I mean, there are other technical questions, but I think you kind of answered that you basically just have a lot of controls, and when you're doing the training set, you probably run the controls all the time when you running the other one, because there is also, when you're doing the 3D reconstruction, you have a stitching issue, that could be influencing, and the tissue, how long it's in the machine, that those are all could contribute to variability. But I think it sounds like you have all those under control.
Yes, and another important thing to do is we of course have a lot of biological knowledge about what we study. And so after measurement, you can do also many validations simply by checking what markers are cells expressing. So if you have a cell that expresses CD3 and CD20, well, I would be rather suspicious about this result. You also know a lot of things about your samples, I think, initially. And so this also gives additional confidence. But as I said, you need lots of controls and lots of validation experiments before you can really trust your results. But I think that's true for any type of large-scale analysis and whatever multiplexed analysis you do.
Yeah, and I feel like I keep telling this to people. I think some of the controls we're doing with antibodies should be done with the companies who actually sell the antibodies.
Yes,
because we have almost like, very high failure rate and things recognizing things that are not even really the target. So anyway, but that's another conversation. So another question about the 3D organization of the tumor. Have you seen and the architecture, the 3D architecture correlating with subtype or vascularity of the tumor?
So in the moment, we only generated a handful of 3D models. And so we do not have yet a statistical basis to actually analyze what I was just asked. But this, of course, are very exciting things to look into, whether there's some like this, whether there's 3D features that then really relate to clinical features.
And another question regarding, you mentioned the clustering of the cluster size seems to be correlating with outcome. And it looks like that the bigger patches are better tumors than the more dispersed ones. Is that in some way relates to metastatic behavior? As you know, tumors may be more likely to metastatize in clusters, or is this related to the immune environment also? Are the cluster tumor cells more likely to survive alone because they already suppress the environment, whereas the others may be in bigger patches because they have not completely evaded the immune attack?
Yes, again, these are excellent questions. So one could assume that if you have much smaller clusters of cells and those cells don't like to stick to each other, like in those big patches, that they might perhaps have higher propensity to metastasize. And I mean, they have a poorer survival, and those patients have distant metastasis. So that's a hypothesis that could be drawn. But to really substantiate that, I think we would have to make other panels that really tell us whether or not those cells have a higher metastatic potential, or whether they're in a more kind of partial EMT state or something like that. And for the immune environment, so that's something that we're currently doing, how that differs, because the immune markers that we have in this panel is very coarse. And now we are doing an analysis with a much more focused immune panel. And then I think I will be able to say more about or to answer better this question. But you already have indications, like with these communities that we find among these triple negative patients that are associated to good prognosis. So I'm sure there will be a relationship like that.
And also you mentioned that you're working on combining this with barcoding technology. And I think you could also combine it potentially with genetic markers that would allow tracking of the tumor cells. Either you could do FISH or some in situ PCR kind of things that you could label, and then you could actually track the cells during treatment or during metastatic evolution, and you could see if are they always neighboring each other, or not neighboring each other?
Absolutely, because if all that I've shown you, we assume it's tumor cells, but to really prove it, we should read out some genomic features.
Okay.
So I think you're making an excellent point here.
Mm-hmm. And then a question about, have you looked at more closely about using evolutionary dynamics and figure out what is the driver and how you're planning to use this knowledge to guide the treatment of the patient?
Yes, again, great question. So in the first study that I've shown you, we did think, and we actually did some preliminary analysis where we did some pseudo-time analysis using the single cell data. Mm-hmm. But in a way, we were very reluctant to include this into the publication because this is mixing cells from different patients where we already know that genomically they're different. So, but if you do this, then you do see more stem cell-like cells, progenitor, etc., the more differentiated ones. But I have to admit, I'm not sure how to interpret this since it's cells from, as I just said. So the way how we currently approach this, from the first cohort, after dissociation of the tumors, we kept viable a part of the tumor that was still viable. We froze that away. And based on those samples, we derived patient-derived xenografts. And so now we studied those to better understand the biological, but also the treatment response of the different phenotypes that we have identified in our studies. Mm-hmm. Because to really address this, you need model systems. Mm-hmm. And that's what you're working on.
Mm-hmm. Okay, and one last question relates to, one of the advantages of the in situ technologies is to be able to assess very small lesions like pre-invasive, very early stage tumors. So have you considered, looking into that, like DCIS or even ADH in breast cancer? And also, what is the limit? How many cells do you really need to be able to make statistically sound conclusions?
That's a function of the question and a function of how different, or how different the cells or populations are that you look at. But I wouldn't dare to draw conclusions if it was below a few hundred cells. Mm-hmm. So, but now that's more gut feeling than having a statistical sound analysis. And in regards to DCIS, actually, Mike Angelo from Stanford, he also gave a talk a while ago in your seminar series. They just put a nice manuscript on bioRxiv where they use their multiplexing technique to actually look at DCIS. So in this regard,
I knew the paper, yeah.
Yes, so in this regard, if that's the interest of the person who asked the question, I would recommend to read this manuscript. Okay,
sounds good. So thank you again for the great talk and a beautiful slides that we could see. And thanks everyone for joining and insightful questions, and looking forward to seeing everyone next Tuesday. So thank you again.
And thank you for inviting me. It's a fantastic seminar series. I watched many of the previous talks. It's really great.
Okay, thank you. Thank you so much.
Bye-bye.